Hello, my name is Carol Leibel, and today we want to examine the process of meiosis and its role in sexual reproduction. Now, there are multicellular organisms that do not employ sexual reproduction, or as an alternative, they have asexual reproduction. And you may ask yourself, well, what's the advantages of asexual reproduction over sexual reproduction? Well, for one thing, it eliminates the need to find a mate. Also, these organisms may reproduce at a faster rate and produce larger number of offspring, using less overall energy and resources. Most scientists believe that asexual reproduction is better in an environment that's stable and is unchanging. However, most places on this planet, that's not the case. And asexual reproduction only employs the process of mitosis. Well, I want to look at a few examples of asexual reproduction. What you're looking at right here is a picture of a hydra, and as you can see right over here, it looks like it's forming a little baby hydra, if you will. And that's exactly what's going to happen. There's a mitosis is going on, and right over here, this little baby hydra, if you will, is growing, and eventually it's going to fall off on the ground and produce another hydra, and that's called budding. Another form of asexual reproduction can be found in sponges, and sponges will produce a specialized mass of cells called a gemmule, and these gemmules can actually overwinter in the late summer and fall, and then in the springtime it'll start to do mitosis and make an adult sponge. Another example is fragmentation. Now, fragmentation occurs when the body of a parent breaks into distinct pieces. Each one can produce an offspring. Turns out that planaria are famous for this, and right over here you can see that happening. Now, another form of asexual reproduction is regeneration, and to be quite honest, it's very similar to fragmentation, and quite often the two are just lumped together. But in regeneration, if a piece of a parent is detached, then it can grow and develop into a completely new individual. So, somewhere in the Orient, fishermen would, as they're looking for oysters, which contain pearls, they noticed that starfish had the ability, or they would eat oysters. So if a fisherman caught a starfish, it would cut that starfish up into several pieces, thinking that it killed the starfish, and throw it back into the ocean. What a mistake that is, because each one of those pieces could regenerate and form a new starfish. As you can see right here in this picture, this is an arm of a starfish thrown back into the ocean, and look right here, it's forming a starfish. Here is a crazy form of asexual reproduction. It's called parthenogenesis, and it's a form of asexual reproduction in which growth and development of the embryos occur without fertilization. So in this particular group of whiptail lizards, there are no males. There are just eggs, and from that egg you will get another lizard. So there are no males in this group. So at any rate, it's kind of amazing to me, if you will. Alright, so we saw some forms of asexual reproduction. It seems pretty straightforward. But what about sexual reproduction? I mean, sexual reproduction certainly uses a lot more energy and resources and takes a longer time, but there must be some distinct advantage of sexual reproduction over asexual reproduction, that it's so dominant in our life. Well, the advantages include that you can have a varied number of phenotypic offspring. They're going to be different than their parents. some of those differences may indeed be better for the environment and give them an advantage. Some of those differences may actually be at a disadvantage. Sexual reproduction involves the union of two haploid cells from different parents to produce a diploid zygote. And so in this process of sexual reproduction, somewhere along the line, meiosis must take place. So you're looking at two different Hollywood families. This one right here is Sissy Spacek and her two daughters and husband, and you can see some similarities, but yet there are indeed some differences. What you're looking at over here is the family of Lloyd Bridges and his two sons and daughter, and again you can see some similarities, but there are indeed some differences. So we want to look at some different life cycles that employ sexual reproduction. So there's some definitions that we have to go over. Alright, the first definition is the term haploid. Now, a haploid is a cell or a unicellular organism that has just one complete set of chromosomes. It's not unusual to see certain species of fungi that are actually haploids. Now, a diploid, and we are indeed a diploid, is an organism that has two complete sets of chromosomes. So we actually have 23 homologous pairs of chromosomes, or a total of 46 chromosomes. So we want to look at a diploid life cycle. So a diploid life cycle means that as an adult you have two sets of chromosomes. When it comes time to reproduce those, somehow, some way, you have to have produced cells that are indeed haploid. So if you're looking at humans, and we are indeed diploids, in our reproductive organs, we are going to produce, through the process of meiosis, egg cells and sperm cells. And then in the process of reproduction, fertilization is going to take place, and we're going to restore the diploid number to N. And when you have a fertilized egg, by the way, that's called a zygote. And once you have that little fertilized egg, you and I both know what's going to happen. Mitosis, mitosis, mitosis. And we're going to end up producing an infant, if you will. Now, the advantage of being a diploid is that deleterious genes can be masked if they're recessive. So you have two sets of chromosomes. Chances are you have some bad genes, if you will, and they may be recessive. But you never know that you have those bad genes or deleterious genes, if you will, because you have another set of chromosomes that covers it up. However, there is a disadvantage to being a diploid, and that is you have more chromosomes to manage. And there's an increased likelihood that something is going to go awry. Alright, so got a diploid life cycle? La-da-da-dum! We have a haploid life cycle. So, as I mentioned before, there are many species of fungi that are haploid. And so what happens is during their lifetime they do mitosis, 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 and by the way, they grow strings of cells called hyphae. When the conditions are absolutely perfect, what will happen is that these two haploid hyphae will each donate nuclei, and fertilization will take place. And now you have a diploid condition, a zygote. But it doesn't stay a zygote or a diploid very long. What will happen very quickly is that meiosis will take place, and it will then produce haploid spores. Those haploid spores will then fall to the earth, and reproduction will take place. So meiosis takes place immediately after fertilization. The disadvantage of being a haploid is that what you see is what you get. You can't mask any bad genes, if you will. However, the advantage of being a haploid is that it's easier to reproduce with fewer chromosomes, and you decrease the likelihood that something is going to go awry. And last but not least, we have a third different life cycle, and that involves plants. Now, it sounds kind of crazy, but it's called the alternation of generation. And what that means is that if I'm a diploid, my offspring are going to be haploid. And if I'm a haploid, then my offspring are going to be diploid. And the two generations do not look like one another at all. And so it alternates between a haploid and a diploid state. Now, in botany, they give those generations special names. If you're the diploid generation, you're called the sporophyte generation. And if you're the haploid generation, you're called the gametophyte generation. So let's look at what happens. Let's suppose we're talking about an apple tree. Now, an apple tree is the sporophyte generation. It's diploid. You know that apple trees, they have flowers, and they produce pollen. Now, in that process of the flowers, you're going to have the process of meiosis taking place, and you're going to produce little spore-like spores called microspores and megaspores, and they're going to do mitosis, mitosis, mitosis. So instead of producing eggs and sperm right away, what you're going to do is you're going to produce a male gametophyte and a female gametophyte. Now, a male gametophyte is nothing more than a piece of pollen, and the female gametophyte remains protected in an ovary inside the flower. Now, the male and the female gametophytes, now those are the ones that are going to do mitosis. And they're going to produce, inside the little female gametophyte, she's going to produce an egg. And inside that piece of pollen, there will be 2 sperm cells. And what will happen is the sperm will fertilize the egg, and then you'll get your zygote. Now, mitosis, mitosis, mitosis will take place, and we restored the diploid generation, and we're going to go ahead and get a seed found inside the apple. But at any rate, you do not need to know the specifics, but you need to know that indeed plants undergo something called the alternation of generation. And this is kind of interesting during the course of evolution, because what we have seen over time is a change in the importance of these two gametophyte and sporophyte generation. So if you're looking at mosses and other non-vascular plants, the most obvious or dominant generation turns out to be the gametophyte generation. So when you look at something and it's a moss or a moss on a tree or on a rock, those are gametophytes. The sporophyte generation is very inconspicuous. And what happened over time is we see now there's an increase in the sporophyte generation. It becomes more obvious, more conspicuous, and the gametophyte generation becomes smaller. And we see this happening over time, so that the more dominant conspicuous generation becomes the sporophyte generation, the diploid generation, and the more inconspicuous or smaller generation is the gametophyte or the haploid generation. Alright, so let's look at a diploid cell. You may be asking yourself, how in the world did you become diploid? That seems like an obvious answer. During the process of fertilization, you got one set of chromosomes from your father and another set from your mother. So what we're looking at right here is a cell, and you'll see that these 2 chromosomes right here, they're different colors because one came from one parent and one came from the other parent, but they are the same length and they had the same genes on them in the same order. Now, the form of those genes or the alleles may be different. Perhaps like right here is the location where you would find eye color. And this one you got from your father may be for brown eyes, but this one over here that you got from your mother may be blue eyes. Alright, and so these two chromosomes are the same chromosomes and these two over here. The reason that these chromosomes are double-stranded because they've gone through the process of DNA replication or synthesis, the S phase. So what you're looking at right here is a double-stranded chromosome with two chromatids. Now, what we would also say is that 2N is equal to 6 because there are 6 chromosomes there. And one set of chromosome, or N, is equal to 3. And that we got one set from the mother, so that's the maternal set of chromosome. We got one set from the father, and so that's the paternal set of chromosomes. And so therefore we have 2 sets of chromosomes. These two right here, because they have the same genes in the same order, they're said to be a homologous pair of chromosomes. So we have 3 homologous pairs of chromosomes in this little diagram that we're looking at. Now, one of the things that they have done is that they can take a picture of your chromosomes and they can match them up. And that's called a karyotype. Humans have 23 pairs of chromosomes in their somatic cells, and in this case here, they usually use white blood cells, and they have the chromosomes have been stained, and what we see is a certain banding pattern. And they were literally cut out, these little chromosomes, and they will try to match them up to one another. As we've already said, 2 chromosomes that have the same genes in the same order are said to be homologous chromosomes, and in a karyotype they should have the same banding pattern. They'll have the same length, and as we mentioned, carry the same genes. Here is another karyotype, and they have already gone through the process of matching up the chromosomes. So this here, that little square, represents chromosome number 1, and then 2, 3, 4, and so forth. Down here at the bottom, you can see that there's an X and a Y, and they do not match up. Now, it turns out there are a few genes they have in common, but there are many genes on there that they do not have in common. And those are called our sex chromosomes. Autosomes are the non-sex chromosomes. So we have 22 pairs of autosomes and 1 pair of sex chromosomes. Alright, so just to give you an overview of what's going to happen during the process of meiosis. Meiosis is a special type of cell division, and it's going to produce 4 haploid cells from 1 diploid cell, and these haploid cells are going to be genetically different from one another. In the process of meiosis, there are actually 2 cellular division events, meiosis 1 and meiosis 2. Meiosis 1 is going to produce 2 haploid cells with double-stranded chromosomes. Each chromosome, of course, has 2 chromatids that are going to be genetically different from one another. And then the whole purpose of meiosis 2 is to separate those chromatids and to produce 4 haploid cells that we have separated the hybrid chromatids. Alright, so here's just an overview of what's going to happen in meiosis. Here we have 2N is equal to 4. And the chromosomes are double-stranded, And after meiosis 1 takes place, notice that we have two cells. Each one only has two chromosomes. So here, n is equal to 2. So what we have done is we've gone from being a diploid to a haploid, and the chromosomes are genetically different from one another. So now let's look at what's going to happen in meiosis 2. So here was meiosis 1. But in meiosis 2, we're going to separate these chromatids, and they're going to become single-stranded. We still have, this is 2n, this is n, and this is also n. This is equal to 4, this is equal to 2, but they're double-stranded, and this is equal to 2, and they're single-stranded. I want to remind you that cells undergoing the process of meiosis will undergo the process of interphase, and like mitosis, it's composed of G1, S, where the DNA is replicated, and G2. So when meiosis begins, that cell already has double the amount of DNA that it normally would have. So the first phase of meiosis 1 is called prophase, and quite often it's lumped with prometaphase. And so let's look at what's going to happen in prophase, because it's a pretty important event. So first thing that's going to happen is that the chromosomes are going to become visible as long filaments. That makes sense. But now let's look at what happens. Homologous chromosomes somehow are going to find one another. This did not happen in mitosis. And they're going to pair and align as long, well-separated filaments. And then something bizarre is going to happen. Recombination is going to occur. So let's look at this. Okay, this is being summarized here in this little animation.
Prophase 1 begins with condensation of the chromosomes. Homologous chromosomes, each made up of two sister chromatids, come together in pairs. This pairing is called synapsis. Each chromosome pair is called a tetrad, a complex of 4 chromatids. Chromatids of homologous chromosomes cross over each other and exchange parts. Meanwhile, other cellular components prepare for the division of the nucleus. The centrosomes move away from each other and spindle microtubules form between them. The nuclear envelope and nucleoli disperse. Finally, spindle microtubules capture the chromosomes, and the chromosomes begin moving to an imaginary plane equidistant from the poles.
Alright, so let's look at what happens during this process of synapsing. So here are two homologous chromosomes, and they have combined, and they're going to form something called a tetrad, and at exactly precise locations there's going to be a protein come along and clip the DNA so that we get an exchange between the two chromosomes. Now the places where crossing over or that clipping and recombination is taking place is called a chiasma. So in this particular diagram there are two chiasmas. Okay? When they're formed together as a tetrad, it's called a synaptomenal complex. And it turns out that there is a protein holding those two chromosomes, homologous chromosomes, together. That protein is called ZIP1. And there, as I mentioned, there's a large recombination nodule that's going to come along and clip the DNA on both sides of the chromatids and recombine them to form hybrid sister chromatids. Now, They also are going to have a centromere. However, and a kinetochore. However, the microtubules are only going to attach to one side of the kinetochore as opposed to both sides. So when you separate, when separation takes place, instead of separating chromatids, you're going to separate homologous chromosomes. Here's another diagram showing you how a tetrad is formed. Here are the homologous chromosomes. Here are the centromere area with the kinetochores. Okay. Here is the synaptomental complex, and again, there's a protein called ZIP1 holding them together. And then now here's the recombination nodule coming down and clipping it at very specific places so that you end up with hybrid sister chromatids. And it's kind of interesting because we always show that the chromosomes are side by side, but in reality, one actually lies on top of the other. Well, let's look at the effects of crossing over on genetic variation. If you have no crossing over and meiosis takes place, after meiosis 2, you're going to have 4 chromosomes, and 2 of these are going to be identical, and these 2 will be identical. But if you have crossing over taking place, look what happens. All 4 of these chromosomes are different from one another, and these are called hybrid chromosomes, if you will. These 2 right here in the middle have bits and pieces from one another. They're recombinant, if you will. And this is important in promoting genetic variation. Alright, so let's look at this process of crossing over in this little animation.
Now let's look at how crossing over... Now let's look at how crossing over creates even more genetic variability. During prophase 1 of meiosis, homologous chromosomes pair up very closely, and corresponding parts of 2 non-sister chromatids may trade places. This process of crossing over creates variation by producing chromosomes that combine the genes inherited from 2 parents. Here the process produced a total of 4 genetically different gametes. There are many ways crossing over can occur. Note that crossing over produces some parental gametes with chromosomes like those of the parents, and some recombinant gametes with a mixture of genes from both sets of chromosomes.
As I've mentioned before, the kinetochore microtubules only attach to one side of the kinetochore. So we're going to separate homologous pairs of chromosomes instead of separating the chromatids during meiosis 1. Alright, so metaphase 1 of meiosis 1. After the crossing over takes place, the microtubules tug back and forth, and eventually the homologous chromosomes move along a unit to the metaphase plate. Each homologous chromosome has a different kinetochore microtubules attached to the centromeres. Now the question is, which side of the plate do they end up on? So here, these 2 chromosomes, the maternal is on one side and the paternal is on the other, but this one right here, it's just the opposite. And that has huge implications on what kind of gametes you're going to make. So what determines which side of the metaphase plate you end up on is just a matter of chance. It's kind of like flipping a coin, but it is extremely important in promoting genetic variability, just like crossing over did. Alright, so let's look at how this affects the different kinds of gametes. So here we have 2 chromosomes, and we've lined them up so that all the parental are on one side and all the maternal are on the other side. We have meiosis 1 taking place right here, and here's the result. And now here are the gametes. And you can see that let's not worry about crossing over right now. Let's assume no crossing over took place. It's a very unusual situation. So what we see here is that these two gametes are going to be different from these two gametes. But what if you had a change and one of these pair of homologous chromosomes lined up differently on the metaphase plate? Look at what happens to the gametes. Combination number 3 is different from combination 1 or 2, and again different from combination number 4. So this business of lining up on the metaphase plate does affect the type of gametes that you're going to produce. So we only had 2 homologous pairs in this example. What would it look like if you had 3 pairs? Alright, so here, look, we have 3 pairs shown right here. So you could line up like this. This is situation number 1. This is situation number 2. This is situation number 3. This is situation number 4. There are no other combinations in terms of lining up on the metaphase plate that you could produce. And once we go through meiosis 1 and meiosis 2, you can see we're going to produce not 4, but 8 different types of gametes. And if you think about it, the chromosomes have 2 different ways they can line up. They can line up like that, or they can be just the opposite. So there are 2 choices, okay? And then now, how many different pairs do you have? In this case here, we have 3 different pairs, so it's 2 to the 3rd, and that's how we get 8 different combinations. Now, humans have 23 pairs of chromosomes. So if we want to know how many different combinations you could get with humans, well, first off, you still have 2 ways a chromosome can line up, but we have 23 pairs. Oh my gosh, 2 to the 23rd. It's an incredible number of different kinds of gametes you could make. It is somewhere on the order of different kinds of gametes. And I gotta say, I don't have that many eggs, but it does make sense why your sisters and brothers, your siblings, look so very different from you unless you're identical twins. Wow, that is just amazing. Alright, so let's look at this little animation about metaphase.
At metaphase 1, spindle microtubules from one pole of the cell are attached to one homologous chromosome, while spindle microtubules from the other pole of the cell are attached to the other chromosome of the pair. Thus, the homologous chromosomes are poised to move to opposite poles of the cell.
Alright, so after metaphase 1, as we've mentioned, you're going to separate homologous pairs of chromosomes. And that's what happens in anaphase 1. The centromeres do not separate. Homologous chromosomes do separate, and they're going to move towards the opposite poles. The spindle fibers interact to force those poles apart. Non-disjunction is a case where the homologous pairs of chromosomes do not separate like they're supposed to. And if that doesn't happen, well, it turns out then you're going to have a mistake in the number of chromosomes in those daughter cells. So this can happen if a tetrad do not separate properly during meiosis 1, and they have found a few cases where the sister chromatids may fail to separate during meiosis 2. So in this situation right here, we have these two chromosomes, okay, and they are supposed to separate so that each one gets 2, but you have non-disjunction here, and so what happens is this particular gamete ends up with 3 chromosomes and this one only ends up with 1. We have some examples of non-disjunction, and one little example happens in females, in which females actually are missing one of their X chromosomes, and that is called Turner syndrome. And in Turner syndrome, the females are usually smaller in stature. Unless they get medication, they don't go through the process of puberty. Their IQ is just fine. However, given medication and hormones, they can be normal. However, quite often they will be sterile. So now, if you have a gamete that doesn't have the correct number of chromosomes, like shown in this picture, so this one has an extra chromosome, this one has an extra chromosome, and this one is missing one, you can end up with a gamete that doesn't, a zygote that doesn't have the correct number of chromosomes, and that's called aneuploidy. The classic, let's backtrack. The classic example of aneuploidy is called trisomy 21, where there's an extra number 21 chromosome. So instead of having 46 chromosomes, they have 47. And individuals who have trisomy 21 usually have a lowered IQ, a rounded mouth with protruding tongue, almond-shaped eyes, and heart defects. We usually find trisomy 21 in older parents. Alright, so here's a little animation to talk about anaphase.
During anaphase 1, each pair of chromosomes is pulled apart, and the homologous chromosomes move toward opposite poles. Note that sister chromatids remain attached at their centromeres and move as a unit toward the same pole.
Alright, so let's move on to telophase 1. The chromosomes, as we see, are still double-stranded, and they contain 2 hybrid chromatids. They've migrated to the poles. The nuclear membrane is going to reform, cytokinesis is going to take place, and we're going to end up with 2 haploid cells. However, the chromosomes are double-stranded. So let's look at this little animation about telophase 1.
Producing 2 haploid daughter cells. The chromosomes finish their journey during telophase 1, and cytokinesis occurs, producing 2 haploid daughter cells. Note that each chromosome still consists of 2 sister chromatids. Meiosis isn't over yet. Remember that it consists of 2 consecutive divisions. During meiosis 2, the sister chromatids will be separated.
Alright, so now we're ready to begin meiosis 2. But the time between meiosis 1 and meiosis 2 is called interkinesis. And there's a big difference between interkinesis and interphase. And that big difference is that no DNA replication occurs. The chromosomes are already double-stranded, so that's not necessary. Meiosis 2 is almost exactly like mitosis, except for the fact there's no DNA replication, and the whole purpose of meiosis 2 is to separate these double-stranded chromosomes. Okay? Interkinesis, we just went over that. It's the time between meiosis 1 and meiosis 2. The big important thing is no further DNA replication occurs because it's already double-stranded. So now let's look at prophase 2. The centrosomes have replicated and they begin to move apart. The chromosomes appear as long, thin threads. The nucleolus becomes less distinct. Asters begin to form. Twin chromatids become visible. The kinetochore microtubules are going to attach to both sides of the kinetochore. Okay, and the spindle is going to form and the membrane begins to disappear. So again, here's a nice little diagram showing you the big difference between prometaphase 1 or prophase 1 versus prometaphase 2. And as you can see, only one side, the spindle fiber is only attached to one side of the kinetochore. But in prometaphase 2, they're going to attach to both sides of the kinetochore. And we're going to separate homologous pairs of chromosomes. Here we separate chromatids to form single-stranded chromosomes. Well, the microtubules go through the same tugging and pulling that occurs in mitosis in metaphase, and the whole purpose is to line them up on that metaphase plate and to separate, eventually to separate those chromatids. As we have mentioned many times now, the microtubules attach to both sides of the kinetochore, and we have overlapping fibers to push the poles away. In anaphase, the centromeres have separated and the single-stranded chromosomes are now separating and they're moving towards opposite poles. Overlapping spindle microtubules from the opposite poles are interacting to pull the poles apart, and then cytokinesis can begin. In telophase, the nuclear membrane is going to form. The chromosomes begin to uncoil and become chromatin. It becomes thinner. The nucleolus becomes less distinct, and cytokinesis is nearly complete. In the end, we're going to have 4 haploid cells. Alright, so let's look at meiosis 2 and cytokinesis in this little animation.
The second division of meiosis, meiosis 2, is essentially the same as mitosis. The important thing to remember is that meiosis 2 begins with a haploid cell. During prophase 2, a spindle forms in each cell and the chromosomes move toward the middle of each cell. During metaphase 2, the chromosomes align in the middle of each cell. Sister chromatids are pulled apart in anaphase 2. In telophase 2, nuclei form at opposite poles of each dividing cell, and cytokinesis splits the cells apart. Meiosis has produced 4 haploid cells, each with one set of chromosomes.
Alright, so one thing I would like to mention is the process of forming gametes in humans. Spermatogenesis is the production of 4 sperm cells from a primary spermatocyte. As you can see in this little diagram, here's the primary spermatocyte. Meiosis 1 takes place and we have 2 secondary spermatocytes, and then meiosis 2 takes place and we have our spermatocytes that are going to mature and form 4 equalized sperm cells. But look what happens in oogenesis. You have a primary oocyte, but when meiosis 1 takes place, one cell gets the majority of the cytoplasm, the other cell does not. And that forms, that little tiny cell is called a polar body because nothing's going to come of it. Meiosis 2 will take place, and again you're going to have one cell getting the majority of the cytoplasm. And then finally, you're going to end up with one functional egg. These three cells are not going to make it. This happens in other organisms, including plants. Alright, so here's a nice little overview of meiosis.
First, the cells from the testes contain the same amount of genetic material found in other body cells. Through a series of cell divisions called meiosis, that amount is cut in half. Finally, the cells become sperm. Well over 100 million sperm cells are normally ejaculated at once. Each of them contains half the genetic information required to make a human being. Once inside the vagina, the sperm are moved along by muscular contractions of the woman's reproductive organs, and under their own power. Their tails propel them forward.
Alright, so that is spermatogenesis. Here is a human egg, or ovum if you will, and those are the three little polar bodies. And here is a nice review of meiosis.
Humans produce gametes, eggs and sperm, through the process of meiosis. Here, we'll follow the production of male gametes by focusing on this cell as it goes through meiosis. Let's begin in the nucleus, where genetic information is stored in chromosomes. Most of a person's cells are diploid, with two sets of chromosomes. One set is from their mother, shown here in red, and the other set is from their father, shown in blue. Each maternal chromosome has a corresponding paternal chromosome. These matched pairs are called homologous chromosomes. During interphase, chromosomes are duplicated. Each chromosome now consists of two identical copies called sister chromatids. Zooming in, we see that each sister chromatid is made up of DNA wound around histone proteins. Each strand coils up into a tight helical fiber. As meiosis begins, a spindle forms and duplicated centrosomes start to migrate toward opposite poles of the cell. Back in the nucleus, the chromosomes are condensing. In meiosis, homologous chromosomes stick together in pairs. The close association of homologous chromosomes allows segments of non-sister chromatids to trade places. This recombination of maternal and paternal genetic material is a key feature of meiosis. After the spindle forms and the nuclear envelope breaks down, microtubules from opposite poles attach to each chromosome of the homologous pair, resulting in a tug-of-war. At metaphase 1, the chromosome pairs are positioned in the middle of the cell. The next stage begins when homologous chromosomes separate from each other and move toward opposite poles. Each chromosome still consists of two sister chromatids. This cell began meiosis with 46 chromosomes, but each daughter cell now has only 23 chromosomes. In meiosis 2, microtubules from opposite poles attach to the chromosomes, which then move to the center of the cell. Next, the sister chromatids separate, becoming full-fledged chromosomes that move to opposite poles. Nuclear envelopes reform, and each daughter cell divides into two cells. We started with a single diploid cell, and now that meiosis is complete, we have 4 haploid cells, cells with a single set of chromosomes. These haploid cells mature into gametes that can contribute to the next generation.
Alright, so one of the things you need to be sure you are capable of doing is comparing mitosis with meiosis. So in terms of DNA replication, both of them have DNA replication before the process begins during S. In mitosis, we only have one cellular division event. In meiosis, there are 2. Synapsing of the homologous chromosomes does not occur in mitosis, but occurs during prophase 1 during crossing over between non-sister chromatids. Okay? The number of daughter cells in genetic combination. Mitosis will produce 2 daughter cells that are genetically identical to one another and also to the mother cell. And if you started out being a diploid, your daughter cells will be diploid. If you start out being a haploid, your daughter cells will be a haploid. If you started out being a triploid, your daughter cells will be triploid. Okay? If the process of mitosis is taking place. However, in meiosis, you're going to end up with, you start off with a diploid cell, and you're going to end up with 4 haploid cells containing half as many chromosomes as the original parent cell. And they're going to be genetically different from one another because of crossing over. So if you're talking about animals, mitosis enables multicellular adults to form from a zygote, and mitosis allows cells to be produced for growth, repair, and in some cases asexual reproduction. However, in animals, meiosis is important to produce gametes, to reduce the number of chromosomes by half, and to produce genetic variability among those gametes. Remember, we wrote down how many different kinds of gametes you could possibly make? This is a visual representation of mitosis versus meiosis. And as you can see, in meiosis there are 2 cellular division events, and that we end up with 4 haploid daughter cells from a diploid cell. And in mitosis, you end up with 2 daughter cells, and they are genetically identical to the original parent. So over here, we start off with 6 chromosomes. Each daughter cell has 6, but over here, each daughter cell only has 3. There was once an AP question that asked students to explain where genetic variability arose during the process of reproduction. So the points they were looking for is that you knew about crossing over during meiosis 1 and prophase 1. You knew also about the importance of how the chromosomes lined up on the metaphase plate, and that was random. And the other thing is that there's a random fertilization of gametes. The gametes are so different. The eggs, if you're talking about sperm cells and egg cells, the possibility that you would have 2 identical egg cells being fertilized by 2 identical sperm cells is so remote that you're going to end up with siblings that are going to be different. And also your choice of mates. If you choose a different mate, you're going to produce different offspring. Alright, so that's the end of our examination of meiosis and sexual reproduction from a biological point of view. I hope you enjoyed it, and I hope to talk to you soon again. Take care. Bye.